The European Federation of Biotechnology has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition given by EFP is as follows: the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. The principle of biotechnology, among many, the two core techniques that enabled birth of modern biotechnology are genetic engineering, techniques to alter the chemistry of genetic material, DNA, RNA, to introduce these into host organism and thus change the phenotype of the host organism. Bioprocess engineering, maintenance of sterile microbial contamination-free ambience in chemical engineering processes to enable growth of only desired microbe eukaryotic cell in large quantities for the manufacturing of biotechnological products like antibiotics, vaccines, enzymes, etc. Let us now understand the conceptual development of the principles of genetic engineering. You probably appreciate the advantage of sexual reproduction over asexual. The former provides opportunities of variations and formulations of unique combinations of genetic setup, some of which may be beneficial to the organism as well as the population. Asexual reproduction preserves the genetic information, while sexual reproduction permits variation. Traditional hybridization procedures used in plant and animal breeding very often led to inclusion and multiplication of undesirable genes along with the desired genes. The techniques of genetic engineering, which include creation of recombinant DNA, use of cloning gene, and gene transfer, overcome this limitation and allows us to isolate and introduce only one or set of desirable genes without introducing undesirable genes into the target of organism. Do you know the likely fate of a piece of DNA which has somehow transferred into an alien organism? Most likely, this piece of DNA would not be able to multiply itself in the progeny cell of the organism, but when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. This is because the alien piece of DNA has become part of a chromosome, which has the ability to replicate in a chromosome. There is specific DNA sequence called the region of replication, which is responsible for the initiating replication. Therefore, for the multiplication of any alien piece of DNA in an organism, it needs to be part of chromosome, which has a specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. This can also be called as cloning or making multiple identical copies of any template DNA. Let us now focus on the first instance of the constitution of an artificial recombinant DNA molecule. The construction of DNA recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid. Autonomously replicating circular extrachromosomal DNA of Salmonella typhimurium, Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene. First recombinant DNA emerged from the possibility of linking a gene. First recombinant DNA, Salmonella typhimurium. Plasmid, Salmonella typhimurium, by Stanley Cohen and Herbert Boyer, 1972, isolated antibiotic resistant gene. Stanley Cohen and Herbert Boyer. First antibiotic resistant gene. Plasmid, Salmonella typhimurium. From a plasmid which was responsible for conferring antibiotic resistance, the cutting of DNA at specific location became possible with the discovery of so-called molecular sister restriction enzyme. The cutting piece of DNA was then linked with the cut piece of DNA was then linked with the plasmid DNA. These plasmid DNA act as vectors to transfer the piece of DNA attached to it. You probably know that mosquito act as an insect vector to transfer malarial parasite into human body. In the same way, plasmid can be used as a vector to deliver an alien piece of DNA into the host organism. The linking of antibiotic resistance gene with the plasmid vector became possible with the enzyme DNA ligase, which act on DNA molecule and joins their ends. This makes a new combination of circular, autonomously replicating DNA created in vitro and is known as recombinant DNA. When this DNA is transferred into E. coli, a bacterium closely related to Salmonella, when this DNA is transferred into E. coli, a bacterium closely related to Salmonella, it could replicate using the new host DNA polymerase enzyme and make multiple copies. The ability of multiple copies of antibiotic resistance gene in E. coli was called cloning of antibiotic resistance gene in E. coli. You can hence infer that there are three basic steps in genetically modifying an organism: identification of DNA with desirable genes, introduction of identified DNA into the host, maintenance of introduced DNA in the host, and transfer of the DNA to its progeny. Tools of recombinant DNA technology. Now we know from the foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have key tools, that is restriction enzyme, polymerase enzyme, ligases, vectors on the host. Let us try. Restriction enzyme. In the year 1963, two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of these added methyl groups to DNA and the other cut DNA. The latter was called restriction endonuclease. 1963 were isolated. Restricting enzymes bacteriophage in E. coli for restricting the growth of bacteriophage. The first restriction endonuclease HIND2 whose functioning depend on specific DNA nucleotide sequence was isolated in 1963. Restricting enzymes HIND2 whose functioning depend on a specific DNA nucleotide sequence was isolated and characterized 5 years later. It was found that HIND2 always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. This specific base sequence is known as the recognition sequence for HIND2. Besides HIND2, today we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequences. The convention for naming these enzymes is the first letter of the name comes from the genus and the second two letters come from the species of prokaryotic cell from which they are isolated. Eco R1 comes from Escherichia coli R113. In Eco R1, the letter R is derived from the name of strain. Roman numbers following the names indicate the order in which the enzymes are isolated from that strain of bacteria. Restriction enzyme belong to the larger class of enzyme called nucleases. These are of two kinds: exonucleases and endonucleases. Exonucleases remove nucleotides from the end of the DNA, whereas endonucleases make cuts at specific positions. Each restriction endonuclease functions by inspecting the length of the DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific point and then sugar phosphate backbone. Each restriction endonuclease recognizing a specific palindromic nucleotide sequence in the DNA. First DNA vector. Action of restriction enzyme. The enzyme cuts both DNA strand at the same site. Vector DNA. First DNA vector. GAATTC. The enzyme cut both DNA strands at the same site. Eco R1 cuts DNA between bases G and A only. G and A only is present in the DNA. Eco R1. Second DNA gene of interest. First DNA of the vector. Ligase. DNA fragment join at sticky ends. Recombinant DNA formation of recombinant. Steps in formation of recombinant DNA by action of restriction into nuclease enzyme. Do you know what palindromes are? Steps in formation of recombinant DNA by action of restriction endonuclease enzyme. Do you know what palindromes are? These are groups of letters that form the same word when read both forward and backward. Example millennium. As against a word palindrome, where the same word is read in both directions, the palindrome in DNA is a sequence of base pairs that reads the same way on the two strands, then orientation of reading is kept the same. For example, the following sequence reads the same on the two strands, 5' to 3'. This is also true if reading read in 5' to 3' direction. Restriction enzyme cut the strand of DNA a little away from the center of the palindrome side, but between the same two bases on the opposite strand. This leaves single strand portion. There are overhanging stretches called sticky ends on each strand. These are named so because they form hydrogen bonds with their complementary counterparts. This stickiness of the ends facilitate the action of the enzyme ligase. The enzyme endonuclease are used in gelting to form recombinant molecules of DNA, which are composed of DNA from different sources and genomes. When cut by same restriction enzyme, the resultant DNA fragment have the same kind of sticky ends, and these can be joined together end-to-end using DNA ligases. Foreign DNA. Vector DNA plasmid. Same restriction enzyme cutting both foreign DNA and vector DNA at specific locations. Ligases join foreign DNA to the plasmid. Recombinant DNA molecule transformation. E. coli cloning host. Cells divide. Transformant. Recombinant DNA technology. Action of restriction into nuclease. You may have realized that normally, unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. Separation and isolation of DNA fragments. The cutting of DNA by restriction endonuclease results in the fragment of DNA. These fragments can be separated by a technique known as gel electrophoresis. Since the DNA fragment are negatively charged molecules, they can be separated by forcing to move towards the anode under an electric field through a medium matrix. Nowadays, the most commonly used matrix is agarose, which is natural polymer extracted from seaweeds, red algae. The DNA fragments separate or resolve according to their size through sieving effect provided by the agarose gel. Hence, the smaller the fragment, the farther it moves. Agarose, which is a natural polymer extracted from seaweed, red algae. The DNA fragments separate or resolve according to their size through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, the farther it moves. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide, followed by exposure to UV radiation. Fragment in the visible light and without staining. You can see bright orange colored bands of DNA in ethidium bromide stained gel. You can see bright orange colored bands of DNA in ethidium bromide. Followed by exposure to UV radiation. After staining the DNA with a compound known as ethidium bromide, followed by exposure to UV radiation. Fragments in the visible light and without staining. You can see bright orange colored bands of DNA in the ethidium bromide stained gel. The separated bands of DNA are cut out from the agarose gel and extracted from gel piece. This step is known as elution. The DNA fragment purified in this way are used in the recombinant DNA by joining them with the cloning vectors. Cloning vectors. You know that plasmid and bacteriophage have the ability to replicate within bacterial cells independent of the control of chromosomal DNA. Bacteriophages, because of their high number per cell, have very high copy numbers of their genome within the bacterial cell. Some plasmid may have only 1 or 2 copies per cell, whereas others may have 15 to 100 copies per cell. Their numbers can go even higher. If we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its number equal to copy number of plasmid or bacteriophage. Vectors used are present and engineered in such a way that they help easy linking of foreign DNA and selection of recombinants from non-recombinants. The following are the features that are required to facilitate cloning into a vector. Origin of replication. This is a sequence from where replication starts, and any piece of DNA, when linked to this sequence, can be made to replicate within the host cells. The sequence is also responsible for controlling the copy number of the linked DNA. So if one wants to recover many copies of target DNA, it should be cloned in a vector whose origin support high copy number. Selectable marker. In addition to ORI, the vector requires a selectable marker which helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium, processed in subsequent sections. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, and kanamycin, etc., are considered useful selectable markers for E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. Cloning sites. In order to link the alien DNA, the vector needs to have very few, preferably single recognition sites for the commonly used restriction enzyme. Presence of more than one recognition site within the vector will generate several fragments and will complicate the gene cloning. The ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. For example, you can ligate a foreign DNA at the PAM-H1 site of tetracycline resistance gene in the vector PBR322. The recombinant plasmids will lose tetracycline resistance due to insertion of foreign DNA, but can still be selected out from non-recombinant ones by plating the transformants on tetracycline containing medium. The transformants growing on ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinants will grow in ampicillin containing medium but not on that containing tetracycline. But non-recombinants will grow on the medium containing both the antibiotics. In this case, one antibiotic resistance gene helps in selecting the transformants, whereas the other antibiotic resistance gene gets inactivated due to insertion. The alien DNA and helps in selection of recombinants. Selection of recombinants due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating on two plates having different antibiotics. Therefore, alternative selectable markers which have been developed which differentiate recombinants from non-recombinants on the basis of the ability to produce color in the presence of chromogenic substrate. In this, a recombinant DNA is inserted with the coding sequence of an enzyme beta-galactosidase. The results into inactivation of a gene for synthesis of this enzyme, which is referred to as insertional inactivation. The presence of a chromogenic substrate gives blue-colored colonies if the plasmid in the bacteria does not have an insert. The presence of insert results into insertional inactivation of the beta-galactosidase gene, and the colonies do not produce any color. These are identified as recombinant colonies. Vectors for cloning genes in plant and animals. You may be surprised to know that we have learned the lesson of transferring genes into plant and animals from bacteria and viruses, which have known this for ages. How to deliver genes to transform eukaryotic cell and force them to do what the bacteria or virus wants. For example, agrobacterium innovations. A pathogen of several dicot plants are able to deliver a piece of DNA known as tDNA to transform normal plant cell into a tumor and direct these tumor cells to produce the chemicals required by the pathogens. Similarly, retroviruses in animals have the ability to transform normal cells into cancerous cells. A better understanding of the art of delivering genes by pathogens in their eukaryotic host has generated knowledge to transform these tools of pathogens into useful vectors for delivering genes of of interest to humans. The tumor-inducing TI plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector, which is no more pathogenic to plants but is still able to use the mechanism to deliver genes of our interest into a variety of plants. Similarly, retroviruses have also been disarmed and are now used to deliver derivable genes into animal cells. So once a gene or DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial plant or animal host, where it multiplies. Competent host for transformation with recombinant DNA. Since DNA is a hydrophilic molecule, it cannot pass through cell membranes. Why? In order to force bacteria to take up the plasmid, the bacterial cells must first be made competent to take up the DNA. This is done by treating them with specific concentration of a divalent cation such as calcium, which increases the efficiency with which DNA enters the bacterium through pores in its cell wall. Recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius heat shock and then putting them back on ice. This enables the bacteria to take up the recombinant DNA. This is not the only way to introduce alien DNA into host cells. In a method known as microinjection, recombinant DNA is directly injected into nucleus of animal cell. In another method suitable for plant cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA. In a method known as ballistics or gene gun. And the last method uses disarmed pathogen vectors, which when allowed to infect the cell, transfer the recombinant DNA into the host. Processes of recombinant DNA technology. Recombinant DNA technology involves several steps in specific sequence such as isolation of DNA, fragmentation of DNA by restriction into nucleases, isolation of desired DNA fragment, ligation of the DNA fragment into a vector, transferring of recombinant DNA into the host, culturing the host cells in a medium at large scale, and extraction of the desired products. Let us examine each of these steps in some detail. Isolation of genetic material DNA. Recall the nucleic acid in the genetic material of all organisms without exception. In majority of organisms, this is DNA. In order to cut DNA with restriction enzyme, it needs to be in pure form, free from other macromolecules. Since the DNA is enclosed within the membranes, we have to break the cell open. To release DNA along with other macromolecules such as RNA, proteins, polysaccharides, and also lipids. This can be achieved by treating the bacterial cell, plant, or animal tissue with enzymes such as lysozyme, bacteria, cellulase, plant cell, chitinase, fungus. You know that genes are located on the long molecule of DNA intertwined with proteins such as histones. The RNA can be removed by treatment with ribonuclease, whereas proteins can be removed by treatment with protease. Other molecules can be removed by appropriate treatments, and purified DNA ultimately precipitates out after the addition of gel ethanol. This can be seen as collection of fine threads in the suspension. DNA that separates out can be removed by spooling. Cutting of DNA at specific locations. Restriction enzyme digestions are performed by incubating purified DNA molecule with the restriction enzyme at the optimal condition for that specific enzyme. Agarose gel electrophoresis is employed to check the progression of a restriction enzyme digestion. DNA is a negatively charged progression of a restriction enzyme digestion. DNA is negatively charged molecule, hence it moves towards positive electrode anode. The process is repeated with vector DNA also. The joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA with a specific restriction enzyme, the cut out gene of interest from the source DNA and the cut vector with space are mixed and ligase is added. This results in the preparation of recombinant DNA, amplification of gene of interest using PCR, thermocycler. PCR stands for polymerase chain reaction. In this reaction, multiple copies of the gene of interest are synthesized in vitro using two sets of primers. This is denaturation, region to be amplified. PCR stands for polymerase chain reaction. In this reaction, multiple copies of gene of interest is synthesized in vitro using two sets of primers. Region to be amplified, DS, double stranded DNA denaturation. Primers are added. Healing, DNA polymerase, Taq polymerase, deoxyribonucleotides. Extension, denaturation, annealing, and extension. 30 cycles amplified 1 billion times. First is denaturation, primer annealing, and extension of primers. Extension of primers. Sets of primers, small chemically synthesized oligonucleotides that are complementary to the region of DNA and the enzyme DNA polymerase. The enzyme extends the primers using the nucleotides provided in the reaction and the genomic DNA as template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times, that is 1 billion copies are made. Such repeated amplification is achieved by the use of a thermostable DNA polymerase isolated from a bacterium Thermus aquaticus, which remain active during the high temperature induced denaturation of double-stranded DNA. Thermus aquaticus. Insertion of recombinant DNA into the host cell or organism. There are several methods of introducing the ligated DNA into recipient cells. Recipient cells, after making them competent to receive, take a DNA present in the surrounding. So if a recombinant DNA bearing gene for resistance to an antibiotic ampicillin is transferred into E. coli, the cells of host become transformed into ampicillin resistance gene resistance cell. If we spread the transformed cells on agar plates containing ampicillin, only transformants will grow. Untransformed recipients will die. Since due to ampicillin resistance gene, one is able to select a transformed cell in the presence of ampicillin, the ampicillin resistance gene in this case is called selectable order. Obtaining the foreign gene product, when you insert an alien piece of DNA into a cloning vector and transfer it into a bacterial, plant, or animal cell, the alien DNA gets multiplied. In almost all recombinant technologies, the ultimate aim is to produce a desirable protein. Hence, there is a need for the recombinant DNA to be expressed. The foreign gene gets expressed under appropriate condition. The expression of foreign gene in host cell involves understanding many technical details. After having cloned the gene of interest and have optimized the conditions to induce the expression of the target protein, one has to consider producing it on a large scale. Can you think of any reason why there is a need for large-scale production? If any protein encoding gene is expressed in heterologous host, it is called a recombinant protein. The cells harboring cloned genes of interest may be grown on a small scale in the laboratory. The cultures may be used for extracting the desired protein and then purifying it by using different separation techniques. The cells can also be multiplied in a continuous culture system wherein the medium is drained out from one side while fresh medium is added from the other to maintain the cells of their physiologically most active log exponential phase. This type of culturing method produces a large biomass leading to higher yield of desired protein. If any protein encoding gene is expressed in heterologous host, it is called recombinant protein. If any protein encoding gene is expressed in heterologous host, it is called recombinant protein. The cells harboring cloned genes of interest grown on small scale in laboratory. The cells can also be multiplied in continuous culture system wherein the used medium is drained out from one side while fresh medium is added from the other to maintain the cells in their physiologically most active exponential phase. This type of culture method produces larger biomass leading to higher yield of desired protein. Small volume cultures cannot yield appreciable quantities of products. To produce in large quantities, the development of bioreactors where large volume 100 to 1000 liters of culture can be processed was required. These bioreactors can be thought of as vessels in which raw materials are biologically converted into specific products. Individual enzymes, etc. use microbial, plant, animal, or human cells. A bioreactor provides the optimal condition for achieving the desired products by providing optimum growth condition, temperature, pH, substrate, salt, vitamin, oxygen. Acid base for pH control. Acid or base for pH control, steam for sterilization, culture broth, flat blade impeller, foam breaker, motor, sterile air. Simple stir tank bioreactor. This is sparged stir tank bioreactor through which sterile air bubbles are sparged. Increase surface area for oxygen transfer. Gas entertainment and entrainment. Bubbles dramatically increase the oxygen transfer area. A stir tank reactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor contents. The stirrer facilitates even mixing and oxygen availability throughout the bioreactor. Relatively, air can be bubbled through the reactor. If you look at the figure closely, you will see that bioreactor has an agitator system. Where is agitator? An oxygen delivery system and a foam control system. We have a foam breaker, steam for sterilization, motor, flat bladed impeller, culture cloth, a temperature control system, pH control system, and sampling ports so that small volume of the culture can be withdrawn periodically. Downstream processing. After completion of the biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing as a finished product. The process includes separation and purification, which are collectively referred to as downstreaming processing. The product has to be formulated with suitable preservatives. Such formulation has to undergo through clinical trials, as in case of drugs. Strict quality control testing for each product is also required. The downstream processing and quality control testing vary from product to product. Count Feb. Biotech, as you have learned from the previous chapter, essentially deals with the industrial scale production of biopharmaceuticals and biological using genetically modified microbes, fungi, plants, and animals. The application of biotech include the therapeutics, diagnostic, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. Three critical research areas of biotechnology are providing the best catalyst in the form of improved organism, usually of microbe or pure enzyme, creating optimal condition through engineering for a catalyst to act, downstream processing to purify the protein or organic compound. Let us now learn how human beings have used biotechnology to improve the quality of human life, especially in the field of food production and health. Biotech application in agriculture. Let us take a look at three options that can be thought of for increasing the food production: agrochemical based agriculture, chemical fertilizer, fungicide, pesticide, insecticide, and herbicides. Agrochemicals are chemical fertilizers, fungicides, chemical fertilizers, fungicides, pesticides, insecticides, herbicides. Organic, biocontrol, biopesticide, biofertilizer, genetically engineered crop-based agriculture. The Green Revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Increased yields have partly been due to use of improved crop varieties, but mainly due to use of better management practices and use of agrochemicals, fertilizers, and pesticides. However, for farmers in the developing world, agrochemicals are often too expensive, and further increase in yield with existing varieties are not possible using conventional breeding. Is there any alternative path that our understanding of genetics can show so that farmers may obtain maximum yield from their fields? Is there a way to minimize the use of fertilizers and chemicals so that their harmful effects on the environment are reduced? Use of genetically modified crops is a possible solution. Plants, bacteria, fungi, and animals whose genes have been altered by manipulation are called GMO. GM plants have been useful in many ways. Genetic modification has made crops more tolerant to abiotic stresses, cold, drought, salt, heat, reduced reliance on the chemical pesticide, pest-resistant crops, insect-resistant, RNA, I and Bt toxin, nematode-resistant plant, pest-resistant crops, RNA, flavor saver tomato, nematode resistant plant, and Bt toxin, insect resistant plant help to reduce post harvest losses, increase efficiency of mineral usage by plants. This prevents early exhaustion of fertility of soil, enhanced nutritional value of food, oryza sativa, golden rice, vitamin E enriched rice. In addition to these uses, GM has been used to create tailor-made plants to supply alternative resources to industries in the form of starches, fuels, and pharmaceuticals. Some of the applications of biotech in agriculture that you will study in detail: production of pest-resistant plants that could decrease the amount of pesticide use. Bt toxin is produced by a bacterium called Bacillus thuringiensis, Bt for short. Bt toxin gene has been cloned from the bacteria and been expressed in plants to provide resistance to insects without the need of insecticide, in effect created in biopesticide. Examples are Bt cotton, corn, rice, tomato, potato, and soya bean. Bt toxin gene. Bacillus thuringiensis, ये तो कभी नहीं बोलता. Bt cotton, corn, rice, tomato, potato, and soya bean. Bt cotton, some strains of Bacillus thuringiensis produce proteins. अच्छा, A है क्योंकि ये पेस्ट रेजिस्टेंट, पेस्ट रेजिस्टेंट. पेस्ट रेजिस्टेंट कौन कौन होता है? बीटी टॉक्सिन, RNA, निमेटोड रेजिस्टेंट, फ्लेवर सेवर टोमेटो. Enhanced nutritional value of food, oryza sativa, vitamin E enriched food. In addition to these uses, GM has been used to create tailor-made plants to supply alternative resources to industries in the form of starches, fuels, and pharmaceuticals. Some of the applications of biotech and agriculture that you will study in detail are the production of pest-resistant plants, which could decrease the amount of pesticide use. Bt toxin is produced by a bacterium called Bacillus thuringiensis. Bt toxin gene has been cloned from the bacteria and been expressed in plants to provide resistance to insects without need for insecticide, in effect created a biopesticide. Examples are Bt cotton, corn, rice, tomato, potato, and soya bean. Bt cotton, some strains of Bacillus thuringiensis produce proteins that kill certain insects such as lepidopterans. Tobacco budworm, armyworm. Corn borer, cotton bollworm. Coleopterans, betons, and dipterans, flies, mosquitoes. Kills lepidopterans, tobacco budworm, armyworm, corn borer, cotton bollworm, corn borer और क्या है, cotton bollworm, lepidopterans, coleopterans, betons, and dipterans, flies, mosquitoes. Kills lepidopterans, tobacco budworm, armyworm, corn borer, cotton bollworm, cotton borer और क्या है, cotton bollworm, lepidopterans, coleopterans, betons, and dipterans, flies, mosquitoes. beetles, dipterans, flies, mosquitoes. TFM. They kill Lepidopterans, tobacco budworm, cotton bollworm, armyworm, corn borer, Coleopterans, beetles, and dipterans, flies and mosquitoes. Beet urine genesis form protein crystals during a particular phase of their growth. These crystals contain a toxic insecticidal protein. Why does this toxin not kill the bacillus? Actually, the beta toxin protein exists in inactive protoxin, but once an insect ingests the inactive toxin, it converts into active form due to alkaline pH of the gut, which solubilizes the crystals. The activated toxin binds to the surface of the mid-gut epithelial cells and create pores that causes cell swelling and lysis and eventually death of the insect. Specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into the several crop plants such as cotton. The choice of genes depend upon the crop and the target pest, as most Bt toxins are insect group specific. The toxins is coded by a gene called Cry1ac named Cry. There are a number of them. For example, the proteins encoded by the genes Cry1ac and Cry2ab control the cotton bollworms. That of Cry1ab control corn borer. Cry1ac, Cry2ab. Cotton bollworms. Cry1ab. Corn borer. Cotton ball destroyed by bollworm. Okay, this one is the one that is destroyed by bollworm and a fully mature cotton ball. Pest-resistant plants. Several nematodes parasite a variety of plants and animals, including human beings. A nematode, Meliotica incognita, infects the root of tobacco plants and causes a great reduction in yield. A novel strategy was adopted to prevent this infection, which was based on the process of RNA interference. RNAi takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of a specific mRNA due to complementary dsRNA, molecule that binds to and prevents translation of mRNA silencing. The source of this complementary RNA could be from an infection by virus having RNA genome or mobile genetic element transposons. Mobile genetic material transposons that replicate via an RNA intermediate. Using agrobacterium vectors, nematode-specific genes, Meliotica incognita, tobacco plant. Using agrobacterium vectors, nematode-specific genes were introduced into a host plant. The introduction of DNA was such that it produced both sense and antisense RNA in the host cell. The two RNAs being complementary to each other form a double-stranded DNA that initiated RNAi, thus silenced the specific mRNA of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from parasite. Most plant-generated double-stranded DNA triggers protection against nematode infestation. Roots of typical control plants. Transgenic plant root 5 days after deliberate infection of nematode, but protected through novel mechanism. Biotechnological application in medicine. The recombinant DNA technological process made impact on the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Further, the recombinant therapeutic do not induce unwanted immunological responses as in case of similar products isolated from non-human sources. At present, about 30 recombinant therapeutics have been approved for human use the world over. In India, 12 of these are presently being marketed. Genetically engineered insulin. Management of adult onset diabetes is possible by taking insulin at a regular intervals of time. What would a diabetic patient do if enough human insulin was not available? If you discuss this, you would soon realize that one would have to isolate and use insulin from other animals. Would the insulin isolated from other animals be just as effective as that secreted by the human body itself and would not elicit an immune response in the human body? Now imagine if bacterium was available that could make human insulin. Suddenly the whole process becomes so simple. You can easily grow a large quantity of the bacteria and make as much insulin as you need. Insulin used for diabetes was earlier extracted from pancreas of slaughtered cattle and pigs. Insulin from an animal source, though, caused some patients to develop allergy or other types of reactions to the foreign protein. Insulin consists of two short polypeptide chains, chain A and chain B, that are linked together by disulfide bridges. In mammals, including humans, insulin is synthesized as a prohormone, like a proenzyme. The prohormone also needs to be processed before it becomes a fully mature and functional hormone, which contains an extra stretch called the C peptide. The C peptide is not present in the mature insulin and is removed during maturation to insulin. The main challenge for production of insulin during our DNA technique was getting an insulin reassembled into a mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced in plasmid E. coli to produce insulin chains. Chain A and B were produced separately, extracted and combined by creating disulfide bonds to form human insulin. Immature insulin, proinsulin, because it has C chain also. C chain, B chain and A chain. A and B has disulfide bonds. C peptide 33 amino acids, A peptide 21 amino acids, A, B, C 21, 30, 33. 21, 30, 33. A amino acids, A peptide 21 amino acids, B peptide 30 amino acids. During maturation, removal of C peptide. यानी कि 33 amino acids निकल गए. A and B disulfide bonds, mature insulin. A peptide के 21 and C peptide के 30. So 51 amino acids. Gene therapy. If a person was born in hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy. In an attempt to do this, gene therapy is a collection of methods that allow correction of a gene defect that has been diagnosed in child or embryo. Here genes are insulated into person's cell and tissues to treat a disease. Correction of a gene defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. The first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. This enzyme is crucial for the immune system to function. The disorder is caused due to the deletion of the gene for adenosine deaminase 1990 ADA deficiency. Four-year-old girl. Clinical gene therapy was given in 1990 to a 4-year-old girl with ADA deficiency. This enzyme is crucial for the immune system to function. The disorder is caused due to deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection, but the problem in both these approaches is that they are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of the patient are grown in culture outside. A functional ADA cDNA using retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the gene isolate from marrow cells producing ADA is introduced into cell at early embryonic stages, it would be a permanent cure. Molecular diagnosis, you know that for effective treatment of a disease, early diagnosis and understanding its pathology is pathophysiology is very important. Using conventional methods of diagnosis, serum and urine analysis, early detection is not possible. Recombinant DNA technology, PCR, and enzyme-linked immunosorbent assay are some of the techniques that serve the purpose of early diagnosis. PCR, presence of pathogen, bacteria, virus, is normally suspected only when the pathogen has produced a disease symptom. In this time, the concentration of pathogen is already very high in the body. However, a very low concentration of a bacteria or virus at the time of symptom of the disease are not yet visible can be detected by amplification of the nucleic acid by PCR. PCR is now routinely used to detect HIV in suspected AIDS patient. It is being used to detect mutation in genes in suspected cancer patients too. It is a powerful technique to identify many other genetic disorders. Autoradiography, a single strand DNA or RNA tagged with radioactive molecule probe. Radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because the probe will not have the complementarity with the mutated gene. ELISA is based on the principle of antigen-antibody interaction. ELISA is based on the principle of antigen-antibody interaction. Infection by pathogen can be detected by the presence of antigens, proteins, glycoproteins, etc., or by detecting the antibody synthesized against pathogens. Transgenic animals. Animals that have had their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. Transgenic rats, rabbit, pig, sheep, cows, and fish have been produced. Although over 95% of all existing transgenic animals are mice, why are these animals being produced? Normal physiology and development. Transgenic rats, rabbit, pig, sheep, cow, fish. Rat, 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 rabbit, pig, sheep, cow, fish. 95% are mice. Why are these animals being produced? How can man benefit from such modifications? Normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functioning of the body and its development. Study complex factors involved in growth, such as insulin-like growth factor, by introducing genes from other species that alter the formation of this factor and studying the biological effects that result. Formation is obtained about the biological role of the factor in the body. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. These are specially made to serve as models for human diseases so that investigation of new treatments for diseases is made possible. Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Specifically made to serve as models for human diseases. Development of diseases. These are specifically made to serve as models for human diseases so that investigation of new treatment for diseases is made possible. Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Biological products. Medicines required to treat certain human diseases can contain biological products, but such products are often expensive to make. Transgenic animals that produce useful biological products can be created by the introduction of the portion of DNA or gene which codes a particular product, such as human protein alpha 1 antitrypsin used to treat emphysema. Similar attempts were being made toward the treatment of phenylketonuria, cystic fibrosis. 1997, transgenic cow Rosie was produced. Transgenic for human disease. कौन सी? Cancer, cystic fibrosis, rheumatoid arthritis, Alzheimer's. Biological product alpha 1 antitrypsin. Alpha 1 antitrypsin for emphysema. Attempts are being made for the treatment of PKU and cystic fibrosis. 1997, first transgenic cow Rosie. Produced human protein enriched milk 2.4 grams per liter. The milk contained the human alpha lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. Vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccines before they are used in humans. Transgenic mice are being used to test the safety of polio vaccine. Vaccine safety in polio vaccine. Polio vaccine. Chemical safety testing. This is known as toxicity or safety testing. This procedure is the same as that used for testing toxicity of drugs. Transgenic animals are made that carry the genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to toxic substances and the effects studied. Toxicity testing in such animals will allow us to obtain results in less time. Ethical issues. The manipulation of living organisms by the human race cannot go any further without regulation. Some ethical standards are required to evaluate the morality of all human activities that might help or harm living organisms. Going beyond the morality of such issues, the biological significance of such things is also important. Genetic modification of organisms can have unpredictable results when such organisms are introduced into the ecosystem. Therefore, the Indian government has set up organizations such as GEAC, Genetic Engineering Approval Committee, which will make decisions regarding the validity of GM research and safety of introducing GM organisms for public services. The modification usage of living organisms for public services as food and medicine source, for example, has also created problems with patents granted for the same. There is growing public anger that certain companies are being granted patents for products and technologies that make use of the genetic material, plants and other biological resources that have long been identified, developed and used by farmers and indigenous people of specific region or country. Example, rice is an important food grain, the presence of which goes back to thousands of years Asia's agricultural history. There are estimated 2 lakh varieties of rice in India alone. The diversity of rice in India is one of the richest in the world. Basmati rice is distinct in its unique aroma and flavor, and 27 documented varieties of basmati are grown in India. There is reference to basmati in ancient texts, folklore and poetry as it has been grown in over 4 centuries. In 1997, an American company got patent rights on basmati rice through US Patent and Trademark Office. 1997, rice patent. US Patent and Trademark Office. This allowed the companies to sell new variety of basmati in the US and abroad. This new variety of basmati had actually been derived from Indian farmers' varieties. Indian basmati was crossed with semi-crop varieties and claimed as an invention or a novelty. The patent extended to functional equivalence, implying that other people selling basmati rice could be restricted by the patent. Several attempts have also been made to patent users, products, and processes based on Indian traditional herbal medicines, example turmeric and neem. If we are not vigilant, we do not immediately counter these patent applications, yet the countries may encash on our rich legacy and we may not be able to do anything about it. Biopiracy is the term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. Most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, use of bioresources by multinational companies and other organizations without proper authorization from countries and people concerned without compensatory payment. Most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, the developing and underdeveloped world is rich in biodiversity and traditional knowledge related to bioresources. Traditional knowledge related to bioresources can be exploited to develop modern application and also be used to save time, effort and expenditure during their commercialization. There has been growing realization of the injustice, inadequate compensation and benefit sharing between developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the Second Amendment of the Indian Patents Bill that takes such issues into consideration, including patent terms, emergency provisions, and research and development initiatives. Because white flowers are more visible in low light conditions. This makes them more attractive to nocturnal pollinators such as moths and bats, which are often attracted to light colored flowers. White flowers may release a stronger scent at night, attracting these pollinators. Our living world is fascinating, diverse, and amazing complex. We can try to understand complexity by investigating process various levels of biological organization, macromolecules, cell, tissue, organs, individual organism, population, communities, ecosystem, and biomes. At any level of biological organization, we can ask people singing every morning. Operation of the voice box, vibrating bone in the bird. The second question is, birds need to communicate with its mate during breeding season. You have already learned in previous classes that ecology is a subject which studies the interaction among organisms and between the organism and its physical abiotic environment. Ecology is a subject which studies the interactions among organisms and between the organism and its physical abiotic environment. Interaction among organisms, between organisms and its physical abiotic environment. Ecology is basically concerned with 4 levels of biological organization: organism, population, communities, and biomes. In this chapter, we explore ecology at organismic and population levels. Organism and its environment. Ecology at organismic level is essentially physiological ecology, which tries to understand how different organisms are adapted to their environments in terms of not only survival but also reproduction. Physiological ecology. Ecology at organismic level is essentially physiological ecology, which tries to understand how different organisms are adapted to their environments in terms of not only survival but also reproduction. You may have learned in earlier classes how the rotation of a planet around the sun and the tilt of axis cause annual variation, intensity and duration of temperature resulting in distinct seasons. These variations, together with annual variation of precipitation, remember precipitation includes both rain and snow, account for formation of major biomes such as desert, rainforest and tundra. Different biomes form due to tilt of Earth axis and rotation of Earth around the sun. Biome distribution with respect to annual temperature and precipitation. Regional and local variations within each biome led to the formation of a wide variety of habitats. Major biomes of India are shown. On planet Earth, life exists not just in few favorable habitats but even in extreme and harsh habitats. Scorching Rajasthan desert, rain-soaked Meghalaya forest, deep ocean trenches, torrential streams, permafrost, snow-laden polar regions, high mountain tops, thermal springs, and stinking compost beds, to name a few. Even our intestine is a unique habitat for hundreds of species of microbes. Tropical rainforest, deciduous forest, sea coast, lush desert. Major biomes of India: tropical rainforest, deciduous forest, desert, and sea coast. What are the key elements that led to so much variation in the physical and chemical condition? The most important ones are temperature, water, light, and soil. We must remember that the physico-chemical abiotic component alone do not characterize the habitat of an organism completely. Ecology is physiological. Ecology at organismic level is physiological ecology, and we must remember that physico-chemical abiotic component. The habitat includes biotic components also, pathogen, parasite, predators, competitors of the organism with which they interact constantly. We assume that over a period of time the organism had, through natural selection, evolved adaptation to optimize its survival and production in its habitat. Each organism has invariably defined range of conditions that it can tolerate, diversity in their resource it utilizes, and distinct functional role in the ecological system. All these together comprises niche. Habitat more than In habitat more than one species can live. Niche, one species. Physically, biotic, abiotic, more invisible. Niche, range of tolerance, activity, requirement, role. Temperature is the most important ecological relevant environment factor. You are aware that the average temperature on land varies seasonally, degrees progressively from the equator towards the poles and from plains to the mountain tops. It ranges from sub-zero level in polar areas and high altitudes to more than 50 degrees in tropical desert in summer. There are, however, unique habitats such as thermal spring and deep sea hydrothermal vents, where average temperature exceeds 100 degrees Celsius. It is general knowledge that mango tree do not and cannot grow in temperate countries like Canada and Germany. Snow leopards are not found in Kerala forest. Tuna and tuna fish are rarely caught beyond tropical latitudes in the ocean. You can appreciate the significance of temperature to living organism when you realize that it affects the kinetic of enzyme and through it the metabolic activity and other physiological function of the organism. A few organisms can tolerate and thrive wide range of temperature. They are called euthermal, and a vast majority of them restricted to narrow range of temperatures, such are called stenothermal. The level of thermal tolerance of different species determine to a large extent their geographical distribution. Euthermal temperature. Euthermal goldfish, American bullfrog. Stenothermal polar bear. Amazonian poison dart frog. Goldfish, American bullfrog. Polar bear, Amazonian poison dart frog. Poison dart frog. Water is another most important factor in the life of organism. In fact, life on Earth originated in water and is unsustainable without water. Its availability is so limited in desert. For aquatic organism, the quality, chemical composition, pH, water becomes important. The salt concentration measured as salinity in parts per thousand is less than 5 in inland waters. Salt less than 5 in inland waters, 30 to 35 in sea, and more than 100 in hypersaline lagoons. Some organisms are tolerant in wide range of salinity uriohaline, but others restrict narrow range stenohaline. Fresh water cannot live in long for sea water because of osmotic problems. Light. Since plants produce food through photosynthesis, a process which is only possible when sunlight is available as source of energy, we can quickly understand the importance of light for living organisms, particularly autotrophs. Many species of small plants, herbs, and shrubs growing in forest are adapted to photosynthesis optimally under very low light conditions because they are constantly overshadowed by tall canopy trees. Many plants are also dependent on sunlight to meet the photoperiodic requirement for flowering. For many animals too, light is important in that they use the diurnal seasonal variation in light intensity and duration photoperiod as tools for timing their foraging, reproductive, and migratory activities. The availability of light on land is closely linked with that of temperature since the sun is the source for both, but deep, more than 100 meters in the ocean, environment has dark inhabitants not aware of existent celestial source. The spectral quality of solar radiation is also important. UV component of the spectrum is harmful to many organisms, while not all the color component of the visible spectrum are available for marine animals living at different depth of ocean. Soil. The nature and properties of soil in different places vary. It is dependent on the climate, the weathering process, whether soil is transported or sedimentary, and how soil development occurred. Various characteristics of the soil, such as soil composition, grain size aggregation, determine the percolation of the water holding capacity of the soil. These characteristics, along with parameters such as pH, mineral composition, topography, determine to large extent the vegetation in any area. This in turn dictates the type of animal that can be supported. Similarly, in the aquatic environment, the sediment characteristics often determine the type of benthic animals that can thrive there. Response to abiotic factors. Having realized that the abiotic condition of many habitats may vary drastically in time, we now ask how do the organisms living in such habitats cope with manifold stresses, but therefore attempting to answer this question, we could perhaps first ask why. Salt. Inland, less than 5 ppt. Sea, 30 to 35 ppt. More than 100 ppt. One would expect that during the course of millions of years of their existence, many species would have evolved a relatively constant internal within the body environment that permits all biochemical reaction and physiological function to proceed the maximal efficiency and thus enhance the overall fitness of the species. This constancy, for example, could be in terms of optimal temperature and osmotic concentration of body fluids. Ideally, then the organism would try to maintain the constancy of internal environment, a process called homeostasis. Despite varying Suppose a person is able to perform best at 25 degrees Celsius, scorching heat of freezing outside. It could be achieved at home. The car while traveling and at a workplace by using air conditioner in summer. His or her performance would always be maximal regardless of weather around him or her. Here the person homeostasis is accomplished not through physiology but by artificial means. Regulate. Some animals are able to maintain homeostasis by physiological, sometimes behavioral also. Constant body temperature, constant osmotic concentration, etc. All birds and mammals are very few lower vertebrates, and invertebrate species are indeed capable of such regulation, thermoregulation and osmoregulation. Evolutionary biologists believe that the success of mammals is largely due to ability to maintain a constant body temperature and thrive whether they live in Antarctica or in Sahara Desert. The mechanism used by most mammals to regulate their body temperature are similar to the ones the humans use. We maintain a constant body by 37 degrees Celsius. In summer, when outside temperature is more than our body, we sweat profusely. The resulting evaporative cooling similar to what happens with a desert cooler in operation. brings down the body temperature. In winter, when the temperature is much lower than 37, you start to shiver. A kind of exercise produces heat and raises temperature. Plants, on the other hand, do not have such mechanism to maintain internal temperature. So regulators are birds, mammals, few lower invertebrates and lower vertebrates, invertebrates, and humans. Regulators. Birds, mammals, few lower invertebrates and vertebrates, humans. Conformers, 99% animals, all plants, small animals, shrews, hummingbirds. Conform, an overwhelming majority, 99% of animals and nearly all plants cannot maintain a constant internal environment. Their body temperature changes with the ambient temperature. In aquatic animals, osmotic concentration of body fluid change with that of ambient air, water osmotic concentration. These animals and plants are simply conformers. Considering the benefits of constant internal environment to organisms, we must ask why these have not evolved to become regulators. Many simples wear it out and resign themselves to suboptimal performance in hot summer months. Thermoregulation is energetically expensive for many organisms. This is particularly true for small animals like shrews and hummingbirds. Heat loss or heat gain is a function of surface area. Since small animals have a larger surface area relative to their volume, they tend to lose body heat very fast, and when it is cold outside, they have to expend much more energy to generate body heat through metabolism. This is the main reason why small animals are rarely found in polar regions. During the course of evolution, the cost and the benefit maintaining internal environment are taken into consideration. Some species have evolved the ability to regulate, but only over a limited range of environmental conditions beyond which they simply conform. If the stressful external conditions are localized or remain only for a short duration, the organism has two other alternatives for survival: migrate, Siberian crane, Kyo, Lado, Bharatpur, Rajasthan. Move away temporarily from the stressful habitat to a more hospitable area. Turn when stressful period is over. In human analogy, Delhi to Shimla during summer. Birds during winter's long distance migration hospitable areas. Every winter, the famous Colorado, thousands of migratory birds from Siberia. Other extremely cold northern regions. Suspend in bacteria, fungi, and lower plants. Various kinds of thick walled spores are formed with the help of suspend. Bacteria, fungi, lower plants, spores, higher plant seeds, deer hibernation, snailfish, estivation, beer is winter sleep, summer sleep, zooplankton, divers. In bacteria, fungi, lower plants, various kinds of thick-walled spores are found which help them to survive in unfavorable conditions. These germinate on availability of suitable environment in high plants, seeds, and some other vegetative products. Structures serve as means to tide over periods of stress beside helping in disposal. They germinate to form new plants under favorable moisture and temperature conditions. They do so by reducing their metabolic activity and going into a state of dormancy. In animals, the organism, if unable to migrate, might avoid the stress by escaping in time. The familiar case of bears going into hibernation during winter is an example of escape in time. Some snail and fish go into estivation to avoid summer-related problems, heat and desiccation. Under unfavorable conditions, zooplankton species in lake and pond known to die pause a stage of suspended development. Adaptation, while considering the various alternatives available to organisms for coping with extreme in their environment, we have seen that in some areas are able to respond through certain physiological adjustments, while others do so behaviorally, migrating temporarily to a less stressful habitat. These responses are also actually their adaptations. So we can say that is any attribute of the organism, morphological, physiological, behavioral, that enables the organism to survive and reproduce in its habitat. Many adaptations have evolved over a longer evolutionary time and are generally fixed. In the absence of an external source of water, kangaroo rat. The kangaroo rat in North American desert capable of meeting all its water through an internal fat oxidation adaptation. Water is a byproduct. Ability to concentrate its urine so that minimal volume of water is used to remove excretory products. Adaptation. North American desert. Many desert plants have a thick cuticle on their leaf surface and have their stomata arranged in deep with sunken stomata to minimize water loss through transpiration. They also have a special photosynthetic pathway, CAM pathway, that enables their stomata to remain closed during daytime. Some desert plants like Opuntia have no leaves. North American. Opuntia. No leaves reduced to spines. Leaves to spines, and photosynthetic function is taken over by flattened stems. Leaf, spine, flat stems, photosynthesis. Then we have mammals from colder climates. Mammals from colder climates have shorter ears and limbs to minimize heat loss. Allen's rule. The polar seas aquatic mammals have thicker layer of fat blubber below skin that act as insulator and reduces loss of body heat. Allen's rule. Then seal, fat, blubber. Some organisms possess adaptations that are physiological, which allow them to respond quickly to stressful situations. If you had ever been to highly altitude, more than 3,500 meter Rohtang Pass, near Manali and Leh, you must have experienced a called altitude sickness. Symptoms include nausea, fatigue, heart palpitation. This is because in low atmospheric pressure of high altitudes, the body does not get enough oxygen. Rohtang Pass. Rohtang Pass, altitude sickness. The body compensates low oxygen availability by increasing red blood cells production, decreasing the binding affinity of hemoglobin, and by increasing breathing rate. In most animals, the metabolic reaction, and hence called the physiological functions, proceed optimally in narrow temperature range in humans, 37 degrees Celsius, but microbes, archaebacteria, hot springs, and deep sea hydrothermal vents, temperature far exceeds 100 degrees Celsius. And we have archaebacteria. A large variety of marine invertebrates. Many fish thrive in Antarctic waters, where the temperature is always below zero. A large variety of marine invertebrates and fish live at great depths in the ocean, where the pressure could be more than 100 times. Marine invertebrates and fishes. Pressure more than 100. More than 100 times. Some organisms show behavioral responses to cope up with. Desert lizard lack the physiological activity that mammals have to deal with high temperature, but manage to keep their body fairly constant by behavioral means. Basking sun absorb heat and desert lizard. Behavioral. Burrowing into the soil to hide and escape from the above ground heat. Population attributes. In nature, we rarely find isolated single individual of any species. Majority of them in group in a well-defined geographical area, share or compete similar or potentially interbreeding and thus contribute to population. Although the term interbreeding implies sexual reproduction, a group of individuals resulting from even asexual reproduction is also generally considered a population for the purpose of ecological study. All the cormorants in a wetland, rats in abundant dwelling, teak wood, trees in forest, bacterium culture plate, lotus plant are some examples of population. You have learned all through individual organism to cope with a changed environment. It is population level the natural selection operates to evolve. Natural selection operates at population level to evolve the desired traits. Population ecology is therefore important. It links ecology to population genetics and evolution. The population has certain attributes. An individual organism does not. Individual may have birth and death, but a population have birth rate and death rate. Per capita birth and death. The rates hence expressed are changes in numbers increase or decrease with respect to members of population. Here is an example. If a pond with 20 lotus plants last year and reproduction 8 new plants are added, the current population to 28. The birth rate is 8 by 20. The 4 individual in a laboratory population 40 fruit flies died. Specific time intervals, say a week, the death rate is 4 by 40 per week, 0.1 individual per week died. Another attribute characteristic of a population is sex ratio. An individual is either a male or a female, but a population has sex ratio. 60% of the population and females and 40% males. A population at any given time is composed of individuals of different ages. If the age distribution percent individual of a given age or age group is plotted for the population, the resulting structure is called age pyramid. For human population, the age pyramids generally show age distribution of males and females in a diagram. The shape of the pyramid reflects the growth status of the population, whether it is growing, stable or declining. Post-reproductive, reproductive and predictive, expanding, stable and declining or in shape. The size of the population tells us a lot about the status in the habitat. Whatever ecological process we wish to investigate in a population, be it outcome of competition with another species, the impact of predator or the effect of a pesticide application, we always evaluate them in terms of change in the population size. The size in nature could be as low as less than 10 Siberian cranes Bharat wetland in a year. Go to millions. Chlamydomonas in a pond. Population size is the population density is generated as n. Need not necessarily be measured in terms of number only, although total number of generally the most appropriate measure of density population. It is in some cases either meaningless or difficult to determine in an area if there are 200 carrot grass Parthenium hysterophorus carrot grass. कैरेट ग्रास का नाम क्या है? पार्थेनियम hysterophorus. but not only single huge banyan tree with large canopy, stating that the population growth of banyan has relative low characterism underestimating the enormous role of banyan that come in such case. Per percent cover or biomass is more meaningful measure of the population size. Total number is again not easily adaptable measure. If a population is huge and counting is impossible or very time consuming, you have a dense laboratory culture of bacteria and a better dish. What is the best measure to report its density? Sometimes a certain ecological investigation, there is no need to know the absolute population density. Relative density serve the purpose equally well. For instance, the number of fish caught per trap is good enough measure of its total population density in the lake. We are mostly obliged to estimate population size directly without actual counting them or seeing them. The tiger census in a national park and tiger reserve is often based on pug marks and fecal pellets. Population growth, the size of a population for any species is not a static parameter. It keeps changing with time depending on various factors including food availability, predation, pressure and adverse weather. In fact, it is these changes in population density that give us some idea of what is happening to the population, whether it is flourishing or declining. Whatever might be the ultimate reason, the density of population in a given habitat during a given period fluctuates due to changes in four basic processes, two of which, natality and immigration, contribute to an increase in population density, and two, mortality and immigration, to decrease. Natality, increase of birth rate during a given period in population that are added to the initial density. Mortality, number of death given a period. Immigration, number of individuals came into the habitat. Immigration, number of individuals that left the habitat. So if n is the population density at time t, okay, nt plus 1 is equal to nt plus b plus I minus d plus e. B is natality plus immigration minus d, mortality, death, birth, death. You can see from the above equation that population or density will increase if the number of death, births plus the number of immigrants is more than number of death plus the number of immigrants. The normal condition, birth and death rate, are the most important factors. The other two factors assuming are special condition. For instance, if a new habitat is just being colonized, or immigration may contribute more significantly than the birth rate. Growth model does the growth of population with time show any specific and predictable pattern? We have been concerned about unbridled human population growth and problem created by it in our country, and it is therefore natural for us to be curious that different population behave same way or some restraint. Exponential growth, resources, food and space availability is obviously essential for the unimpeded growth of a population. Ideally, when resources in the habitat are unlimited, each species has ability to realize fully its innate potential to grow in number. As Darwin observed while developing his theory of natural selection, that the population grows in an exponential or geometric fashion. If you are in a population of size n, the birth rate, not total number but per capita birth, are represented as b, and death rates, again per capita death rates, as d, then the increase or decrease in n during a unit time period t, dn by dt, is equal to b minus d into n. Intrinsic rate of natural increase. Exponential growth. Intrinsic rate of natural increase. Intrinsic rate of natural increase, and is very important parameter chosen for assessing impacts of any biotic or abiotic factor on population growth. To give you some idea, magnitude of R value for the Norway rat R is 0.015. Flower beetle R is equal to 0.12. In 1981, the R value for human population India (205). The above equation describes exponential geometric growth The pattern of the population. The above equation takes a J-shaped curve. Plot n in relation to time. Population density after time t, population density at time 0, intrinsic rate of natural increase. dn by dt Rn b minus d into n. Responses are not limiting. The growth plot is resources are unlimited. Unlimited resources. Population density, logistic growth, no population of any species. Limited resources. This leads to competition between individuals for limited resources. Limited resources, competition. Fittest survive and reproduce. Given habitat has enough resources to support a maximum possible number beyond which further growth is possible. Beyond which no further growth is possible. The limit in nature is called carrying capacity K. Initially a lag phase, phases of acceleration, deceleration, and finally an asymptote reaches the carrying capacity. Sigmoid curve. Burst bird. Logistic growth. dn by dt is equal to Rn k minus n by k. Carrying capacity, intrinsic rate of natural increase. Resource logistic growth model is a more realistic one. Life history variation, population evolved to maximize their reproductive fitness, also called Darwinian fitness, higher R value. Under particular set of selection pressures, organisms evolve towards the most efficient reproductive strategy. Some organisms breed only once in their lifetime. Breed only one, Pacific salmon, fish, and bamboo. Pacific salmon, fish, bamboo. Breed many times, birds and mammals. Okay, some produce large number of small sized offsprings. Large number small offspring. Oysters, pelagic fish, small number, large offspring, big offspring, birds, mammals. Population interactions. Can you think of any natural habitat not inhabited just by single species? There are no such habitats, such as situation and inconceivable. There's one more species on which it can feed. Even in plant species which makes its own food cannot survive alone. It needs soil microbes to break down the organic matter in soil and return the inorganic nutrients for absorption. Then how will the plant manage pollination without an animal agent? It is obvious that in nature, animal, plant and microbes do not and cannot live in isolation but interact in various ways to form a biological community. Even in minimal communities, many interactive linkages exist, although all may not be readily apparent. Interspecific interactions arise from the interactions of population of two different species. They could be beneficial, detrimental, or neutral, neither harm nor benefit. Mutualism and competition. Predation, parasitism, commensalism, amensalism. Both that benefit are mutualism and both lose in competition. Both parasitism, one benefits parasite and predator, and the interaction is detrimental to the other host and prey. The interaction where one species is benefited and other is neither benefit nor commensalism. Amensalism, one is harmed, the other is unaffected. Predation, parasitism, and commensalism. Interacting species live closely together. Predation, parasitism, and commensalism. Interacting species live closely together. Predation, parasitism, commensalism. Species live close together. Predation, predator and prey. Tiger, deer. Tiger, deer. What would happen? Predator, and most probably it is tiger and deer that readily come to our but sparrow eating seed is no less a predator. Sparrow, seed is no less predator. Although animals eating plants are characterized separately as herbivores, they are in broad ecological context not very different from predators. Herbivores, plants. Besides acting as conduits for energy transfer trophic level, predators play other important roles. They keep prey population under control, but prey species could achieve very high population density and cause ecosystem instability. When certain exotic species are introduced into geographically, they become invasive and start spreading fast. Prickly pear cactus, introduced in Australia in 1920s, caused havoc by spreading rapidly into millions of hectares of rangeland. Finally, the invasive cactus was brought under control after a cactus-feeding predator, a moth, from its nest was introduced into the country. Biological control methods adopted in agricultural pest control ability of the predator to regulate. Predators help in maintaining species diversity in a community by reducing the intensity of competition among competing prey species. In rocky interstitial communities of the American Pacific coast, starfish disaster is important predator. When all starfish were removed, more than 10 species of invertebrates became extinct. Starfish disaster. 10 invertebrates became extinct. If a predator is too efficient, overexploits its prey, they might become extinct. Predator will also extinct. Predators in nature are prudent prey species defenses to lessen the impact of predation. Insects and frogs, cryptically colored. Insects and frog camouflage by the predator. Some are poisonous. Solution of prey. Insects, frog, camouflage. Insects, frog, camouflaged, poisonous, and therefore avoided by the monarch butterfly. To predator, bird, special chemical present in its body. Interestingly, the butterfly acquires its chemical during its capillary stage by feeding on a poisonous weed. For plants, herbivores and predators, nearly 25% of all insects are known to be phytophagous, feeding on plant sap and other plant parts. For plants, herbivores are the predators. Nearly 25% of all insects phytophagous. The problem is particularly severe for plants because unlike animals, they cannot run away. Thorns, acacia cactus, most commonly produce and store chemicals, inhibit feeding and digestion, disrupt in reproduction, and even kill it. Calotropis weed, highly poisonous cardiac glycosides. Calotropis, cardiac glycosides. Cattle, goat, plants commercial sale, nicotine, caffeine, quinine, strychnine, opium. Grazers, browsers. Competition, Darwin spoke of the struggle of existence, survival of the fittest, and he convinced that interspecies competition is a potent force in organic evolution. It is generally believed that competition occurs when closely related species compete for same resource limiting. Firstly, totally unrelated can also compete for the same resource. Shallows, South American lake visiting flamingos, resident fishes. Fishes, zooplankton. Secondly, resource need not be limiting for competition to occur. In interference competition, the feeding efficiency of one species might be reduced due to interfering inhibitory presence of other. Competition is better defined as a process in which the fitness of one species, measured in terms of R intrinsic rate of increase, is significantly lower in presence of another species. It is relatively easy to demonstrate in laboratory experiments as Gauss and other experimental scholars. When resources are limited, competitively superior species will eventually eliminate the other species. But evidence for such competitive exclusion occurring in nature is not always conclusive. Strong and persuasive circumstantial evidence does not exist, however, in some cases. Emington tortoise in Galapagos Island. Greater browsing efficiency of goats. Emington tortoise, goat. Competitive race. The occurrence of competition. Another evidence for the occurrence of competition in nature comes from what is called competitive release. A species of distribution is restricted to a small geographical area because of the presence of a competitive superior species is found to expand its distributional range dramatically. Colonel's elegant field experiments show that on rocky sea coast of Scotland, superior barnacle balanus dominates the intertidal area and excludes the smaller barnacle cathamalus in that zone. Okay, wait. This is by Darwin. Darwin. This is by Gauss and Canals. Balinus Cathamalus. Balinus Cathamalus. Gauss' competitive exclusion principle states two closely related species competing for the same resource cannot coexist indefinitely, and the competition inferior one will eliminate. This may be true for resources limiting, but not otherwise. More recent studies do not support such gross generalization about competition, while they do not rule out the occurrence of interspecies competition in nature. They point out that species facing competition might evolve mechanisms that promote coexistence rather than exclusion. One such mechanism is resource partitioning. If two species compete for the same resource, they could avoid competition by choosing different times of feeding or different foraging patterns. MacArthur showed the 5 closely related species. Coexisting. Warblers. 5 species. Warblers. Parasitism, considering that the parasitic mode of life ensures free lodging in metal and meals, it is no surprise parasitism involves so many taxonomic groups from plants to higher vertebrates. Many parasites evolved to be host specific. They can parasite only a single species of host. In such way that both host and parasite tend to co-evolve. If the host evolves special mechanism by rejecting or resisting the parasite has to evolve mechanism to counteract and neutralize them in order to be successful with the same host species. In accordance with the lifestyle, parasites evolved special adaptations such as loss of unnecessary organs, disease of organ suckers to cling on, loss of digestive system, high reproductive capacity. Life cycle of parasite also become one or two intermediate host or vectors to facilitate parasitism of its primary host. The human liver fluke trematode parasite. Human liver fluke trematode parasite depends on two intermediates, snail and a fish. Snail, fish. Malarial parasite. Malarial parasite. Mosquito. Human. Parasites feed on external surface of the host and are ectoparasites. Familiar examples of this group are lice on humans, ticks on dog. Lice, humans, ticks, dogs, marine fish. Marine fish with copepods. Cuscuta. Parasitic plant. Hedge plant has lost its chlorophyll leaves course of evolution. Host plant which it parasitizes. Parasitic. In contrast, endoparasite that live inside liver kidney. Life cycle of endoparasite more complex, extreme specialization. Typical features. Brood parasitism in birds. Brood parasitism. Then an egg of the host, incubate them, resemble the host egg size and color, reduce the chances of host bird foreign ejecting them. Cuckoo and crow. Commensalism, this is the interaction in which One species benefits, other is neither harm nor benefit. Orchid. Growing as an epiphyte on mango branch. And barnacles growing on back of veil. Cattle egret, grazing cattle. Stir up and flush out insects from the vegetation, otherwise might be difficult for egrets to catch an sea anemone that has stinging tentacles and the clownfish. Clownfish that lives among them. Fish gets protection from predators which stay away from these stinging tentacles. Mutualism. Lichens. Fungi, algae. Or cyanobacteria. Fungus and photosynthetic algae or cyanobacteria. Mycorrhiza, fungi, roots of higher plants. The most spectacular pollination. Fig tree. Wasp. Orchids. Bees and bumblebee. Sexual deceit. Ecosystem. Train cycling. Is it biodiversity and conservation or Yeah.